this is for all three of you, I think. It's come in various forms, but the one that, can I vote this up? This one, how important is methylation, do you think, in these rare diseases? And if, Gavin, do you want to start?
So, yeah, I mean, it's very important. So it's something that we're all thinking about. And in our lab, we think about it a lot, but we haven't actually looked at it at all yet. But it's something that we're really keen to be able to do in the future. So things like X-linked disease and being able to characterize the X-linked X inactivation in female carriers, that may be something that's really interesting for our lab.
Yeah, I think that it is so interesting, but also methylation makes sense in the tissue that you are looking for. So, in my case, in my studies, as we are, for example, studying antithrombin deficiency, antithrombin is expressed in liver. So, methylation makes sense if we have samples of from the liver. So in these particular cases, it will be very interesting. However, unfortunately, the samples that we have is peripheral blood. So the limitation is that where are you studying, where are you sequencing your sample?
Yeah.
Yeah.
Yes. Similarly, I would say, especially in some of our families, where when you see the phenotype is really quite dramatic. It's really quite. You can have a wheelchair-bound patient, and you can have a patient who's barely showing a phenotype. So methylation could be of interest to us there.
Thank you very much. Do we have a question from the room? Yes. Can we have another mic, please? Oh, okay. Should we keep one here and yeah?
Yeah.
Thank you for the presentations. I think I forgot who was speaking about the expansions, but okay. Very curious, how sensitive are you able to detect contractions? And is there a mixture, a mosaic nature of those expansions?
I have not come into a situation of seeing contractions in what we've done with the RFC1 in the patients that we've done, but I do believe that this can actually occur in other instances, and so it will be interesting to see whether we can actually see contractions of these. But I've never seen it in the RFC1 being smaller than the reference allele.
Belen, do you also have experience with?
Yeah, our group has also studied this situation, and for us this is very interesting, and this is a problem that happened also with the when you try to do this rapid polymerase chain reaction, that the main polymerase contract the expansion and makes it difficult to study, to diagnose for the clinicians, and that's why nanopore could be a very interesting solution. And however, also in this kind of this technology is also, I think that we are experience also this contraction of this expansion, and maybe it could be larger than expected, than what we see in the nanopore sequencing. So I think that it's a very interesting point, and we are so interested in this type of DNA structure.
Yeah, and just to follow up on that, is experience as well of detecting interrupted repeats as well?
This is something where I believe this complex other repeat could be interrupted repeats, and I think we're going to need something like the long read to get across these regions. but we have found, when I've sort of spoken with people, certain types of motifs do better in terms of the sequencing than others. The AA triple G seems to be a winner, but the other ones are difficult. I think the other thing is that we have used Southern analysis as well, so we were able to do some validation between the long read and having Southern analysis and knowing what the actual sizes were. And we did see concordance there.
Thank you.
Maybe I'll just add from a sort of technical point of view that we've done quite a bit of sequencing of these various different repeats, and we actually have a poster upstairs if anyone's interested. But we clearly see interruptions of repeats. We can count them. We're quite confident we count them well. But we're also quite confident that it's very, very heterogeneous. So there's not like every cell or every bit of genome has the same repeat length. There's definitely something that's happening in the patient, so that we don't expect one answer and one length in all of them. It's a span, and I think that was at some point thought to be a fault of the sequencing. But I don't think we have enough proof now that it probably isn't. There is a mix, and you're not expecting one repeat number. And also we have done some telomere work where we also definitely see interruptions of the repeats that we're quite confident that we're calling correctly.
Thank you. Do we want to have a question from online? Well,
So this is also one that's come in various forms, but first is for Marina. Which tools did you use to call the repeat expansions and the motif analysis?
I would have to refer that to Ira. Ira Deverson is the person who does our sequencing. So if you can find him in the meeting, he would be the person who can tell you what he's done.
Can't find it right now, but I think there was a similar one for you, Gavin. Do you have any specific tools that analysis tools that you'd like to recommend to the audience?
So everything that we're doing at the moment. Sorry, can I grab that? So much of this stuff is kind of proof of concept. So at the moment it's a bit manual, and we're using the basic nanopore workflows. So we use Guppy and Minimap2 and SAM tools to make our BAM files. And then we're kind of manually interrogating the data using IGV. When we do variant calling, we use Claire 3, and then we use WhatsApp for our haplotype generation. So
That's actually the question.
Basic so far.
What do you use for phasing? So
Yeah, so that's we're using the WhatsApp for phasing the haplotype data. Yeah.
should we, do we have another question from the room? Not right now. So
I'll have one from you, Belen. How much depth do you need to confidently detect a structural variation? A sequencing depth, I assume, is meant here.
Yeah, in our case, we have preliminary data that allows us to trust in even 2 reads, because we are going to very candidate selected samples with an intermediate phenotype, with a gene of interest, and we have this is a second stage of the diagnosis of the search of this pathogenic variant. So in cases when you have a candidate gene, even with 2 reads, and even sometimes 1, but it's very, very tricky, because also we have another step, which is the validation by PCR. In the end, the Sanger sequencing is always you have to confirm by another technique what you are seeing. So nanopore sequencing allows us to make this PCR, because then when we saw this SVA insertion, even with 2 reads, we then design the primers in order to validate it. And then in the internal primer with the consensus sequence of nanopore, and then if we amplify, then we trust in that. But from nanopore, as we go to candidate gene, we trust in 2 reads.
And just a follow-up, or kind of different, but how do you map the transposons? So once you get those reads, how do you then map them? No.
When we get the reads, we then perform this de novo assembly, and we search this kind. we compare this element with the all reported SVA or retro element in the reference. However, this is a very, very unknown world of retrotransposons, because in the literature there's and in the reference databases there's there are very low number of retrotransposon reported. So we are now going to all the data that we have of genomic data from nanopore sequencing with the PromethION, and we are now trying to perform this map of retrotransposon already detected from our group, and to make this map of retrotransposon that we are working with, and we are now trying to publish and to get it to the scientific community, because there's a need to characterize these elements, to the polymorphism, which of them are polymorphism, are errors of the reference that we are dealing with. We see insertion that are present in all our samples, so maybe there are errors in the genome of reference. So this is very interesting field.
Do we have a question from the room? Yes, we do.
This is a question for Gavin. Sorry, I think I missed it in your talk. What sample were you using to try and for your RT-PCR when you're saying, because it wasn't really expressed in blood?
Yeah, so we're using blood, and the difference is that when you use traditional methods, so RT-PCR, agarose gel, and Sanger sequencing, you can't see a band on a gel for a non-expressed gene, and so you can't sequence anything from that. But if you take that PCR product from that blank gel and run it on a fungal flow cell, you'll find you've got thousands of reads from thousands of molecules, just not enough to have a visual band on an agarose gel. I think if it was normal practice to run acrylamide gels, you'd see bands from non-expressed genes, but you don't see those on agarose gels, and we think the reason for that is a phenomenon called illegitimate transcription. That's how it's been reported before, is that all genes are transcribed in all tissues at a very, very low level, a basal level of transcription. And that's too low to be able to detect by Sanger sequencing, but not too low to detect by nanopore sequencing. So that's what we're relying on being able to do. And it works. So we've done it on 15 or 20 retina-specific genes, and they all work. So we're pretty confident in our data now.